Welcome back for more physiology. Here we're continuing our story that we started a few lectures ago, which would be taking active transport to build a membrane potential, which we're now going to convert into something that functions, which is the action potential. So in particular, we're going to deal with action potentials as being positive feedback loops. So the refresher. We have the sodium-potassium pump. Its job is to move sodium ions out, potassium ions in. By out, I mean out of the cell, inside of the cell. For every positive charge, there's a corresponding negative charge. Because of the diffusion gradients, or concentration gradients, because sodium wants in, potassium wants out, if you allow them to, they will move. So you have a channel that we call a potassium leak channel, and that actually will allow potassium ions to move from the inside of the cell to the outside. Every time it does so, it leaves behind a negatively charged partner. And because of Coulomb's law, eventually that positively charged potassium ion is not going to want to leave the negatively charged partners left inside of the cell. That pausing point we call the resting membrane potential. Every cell in your body does this. The exception is not all cells know what to do with it. We call those the excitable tissues, and what they generate is called an action potential. Why do we care about action potentials? Because action potentials allow us to have reflexes. So you get this girl, she puts her hand inside of a hot stove, ouch. What she's going to need to do is probably move her hand away from whatever is probably going to hurt her. So what do? You send a signal that says, pull your hand away. Seems to make sense to us. So it turns out to be a multi-part system where we're going to have something that we call a sensory neuron, which is going to send an action potential, which is that little shiny light, into the spinal cord, which will trigger something called an interneuron, which will then tell a motor neuron, hey, muscle, contract. Inside of that one video, we happen to have 4 different action potentials. So what's an action potential? You're going to have to allow a little bit of hocus pocus magic for right now. But what we're going to have are 2 different types of gates. One's going to be a sodium gate, one's going to be a potassium gate. Something is going to make that sodium gate open up. When the sodium gate opens up, sodium is going to move from the outside of the cell to inside of the cell because that's its concentration gradient. When that happens, we're going to add so many positive charges inside of the cell that's going to make the negatively charged inside become positive. When that happens, we went from being negative to becoming positive. We broke the difference between the outside and the inside. Or we could say we broke its polarization, or we undid its polarization. We call it being depolarized. Step 1, sodium ions move into the cell through a sodium gate. The cell becomes depolarized and positively charged on the inside. The next step is that positive state on the inside of the cell is going to make the sodium gate close, and it's going to make a potassium gate open up. When the potassium gate opens up, potassium ions are going to flood out of the cell because their concentration gradient says leave the cell. When they leave, we're removing positive charges from the inside of the cell, and eventually it's going to come back to being negative again. So we're bringing back our polarization, so we call it being repolarized. What we notice in this pattern is an active depolarization will actually trigger another active depolarization, and that will trigger another active depolarization. That is a positive feedback loop because positive feedback loops continue whatever the stimulus is. In this particular case, a depolarization will cause a depolarization, which will cause a depolarization, which will cause a depolarization, which will cause another depolarization. If I were to turn this into a graph, I get a figure that looks something like this. And this has a lot of stuff going on in it, so I'm hoping that you can kind of see the mouse right now. But what you have is our resting membrane potential, something like negative 70 millivolts, whatever. Negative just means the inside's negative. It doesn't really need to be negative. And something is going to trigger this thing to change. And what we're going to get is the membrane potential has become less negative, meaning it's going to approach 0. It's going to move towards positive. There's a certain point at which that sodium gate will open up. There's a certain voltage, a certain potential that will open up the sodium gate. That state is called threshold. And if I can get the membrane potential to reach threshold, automatically the sodium gate will open up and we'll get depolarization, which is sodium ions flooding into the cell. Once we've reached the end of our depolarization, that will cause the closing of the sodium gates, because the positively charged inside will close the sodium gates, and they will open up the potassium gates, which allow the potassium on out. We call that repolarization, until eventually those gates close. The point at which they close turns out to be well beyond our negative 70 millivolts. So they will stay open, but it's only when we get really negatively charged that they say, oh, maybe I should stop. And the result is we get this weird little bump here at the bottom called a refractory period. The state is referred to as being hyperpolarized. We went a little too far. And what that allows for is one directional flow of action potentials, meaning they will go from a start to a finish. You can't go start halfway to the finish and say, and turn it back around and go to the start.